Okay, let's get started. Hello everyone. Welcome to join us this Statistical and AMS's for Health Data Science seminar series. This is organized by the Stats AI group, which is an ASA interest group. The goal is to organize activities that empower statisticians to participate more actively in AI research and leadership. This is the website of the group, and then we will also upload the seminar recordings to this YouTube channel. Before I introduce today's speaker, just to make an advertisement for this conference. This is co-hosted by the group and then also Harvard Stats and Biostats departments. We have a new format and which is also integrated conference journal peer review model for papers. And then there's a call for paper submission and an abstract. The deadline is approaching, which is May 11th. And then here is the website of the conference, so please check. Okay, now it is my great pleasure to introduce today's speaker, Dr. Josh Siegle, senior scientist and a leader of the Electrophysiology group at the Allen Institute for Neurodynamics. Dr. Siegle's work has been central to advancing large-scale electrophysiology, and then he has played an important role in developing open data resources for the neuroscience community. His research focuses on understanding neurodynamics and on how large populations of neurons coordinate across brain regions to support sensation, cognition, and behavior. And then Josh, welcome, and thank you so much for joining us today to share your work at the Allen Institute. We are very excited to have you here. Before I hand it over to Josh, some logistics. Please hold your question to the end of the when Josh finishes his presentation. You can also type in the chat box. I will monitor that. And then after Josh's presentation, I will call your name and then where you raise your hand and I will call name and you can unmute yourself, ask the question. Okay, I will hand it over to Josh. Josh, it's all yours. Thank you.
Great, thank you so much. So thanks for inviting me. I'm excited to talk to you all today. So yeah, as you said, I'm a senior scientist at the Allen Institute in Seattle, where I lead the electrophysiology group. And I think of myself as a scientist first and foremost, but in order to do the best science possible, I have spent a lot of time engineering platforms that expand what we can measure from the brain and the way that we measure things from the brain. So today I want to tell you about one of those platforms called the Allen Brain Observatory, and we'll talk to what we learned, to tell you a bit about what we learned in the course of sharing the data that it produced. So I've tried to frame it for a statistics and machine learning audience. So instead of dwelling too much on what we discovered with this platform, I'll spend most of the time talking about the structure of the data sets, the unique opportunities we created, and the lessons that we've learned about what makes data actually useful to people who didn't collect it. So in many branches of physics and astronomy, scientific progress is inextricably linked to big shared instruments like telescopes, particle accelerators, neutrino detectors, and yeah, it's obvious that no individual lab is going to build the Large Hadron Collider. You can't build the James Webb Space Telescope in your basement. So these are inherently community platforms, and almost all of the data they produce is openly shared, and then gets analyzed by thousands of people, and same measurements end up supporting many different scientific questions. And that model of data collection and sharing has really shaped these fields. It imposes the need for standardization. It makes any claim verifiable by independent teams. They can just go look into the data that's openly shared. And it also means that any data set can be revisited many years later with new methods, which are obviously constantly evolving. So this stands in contrast to neuroscience, which historically has not looked like this at all. It's been more of a cottage industry with individual labs building their own rigs, running their own experiments, and publishing results that are often impossible to reproduce because the data is really idiosyncratic or simply not publicly available. But around 15 years ago, people started asking what an observatory-style platform would look like if it were developed for systems neuroscience. So this cartoon is from a 2012 Nature commentary by Christoph Koch and Clay Reed, who were two of the scientists that spearheaded the Allen Institute's efforts to map neural activity in living brains. The idea was to build a highly standardized industrial scale pipeline for recording activity from the brain and then making all the results publicly available. So it could function as a shared resource, similar to the way that a telescope functions in astronomy. And the core commitments were to standardize protocols, standardize equipment and rigs, large sample sizes, and open release of all the data. And at the time, this was a somewhat radical proposition because it was fairly uncommon for neuroscience labs to share their raw data, and they certainly didn't run their physiology experiments on a large scale. This was a much more boutique operation. And so most of what I'll talk to you today follows from this original framing, and we'll talk about how it actually played out when we tried to implement it. So before I get to the platform itself, I want to orient you briefly to the biology. So at Allen Brain Observatory focused on understanding the mouse visual system, which is diagrammed here. Mice are nocturnal, so for a long time people were kind of skeptical about why you would study vision in mice, but they actually have a surprisingly sophisticated visual system, and the overall structure is quite similar to the visual system of primates, including humans, which is one reason it's been studied so heavily. And this cartoon shows kind of the flow of information through the mouse brain. Light enters through the eye. It gets converted into electrical signals in the retina, and then those signals travel through a small relay structure in the thalamus, here labeled as DLGN. And they also get sent to another structure in the midbrain called superior colliculus, labeled as SC. And then the signals eventually end up in primary visual cortex, labeled as V1. And then from there, they kind of fan out to other visual areas called the HVAs, or higher visual areas, and also loop back through this other thalamic structure called LP, and eventually get distributed much more broadly across the brain. But kind of these are the main structures that form the gateway for vision within the mouse brain. And so the key takeaway from this is don't have to remember what all these different areas are, but you do have to know that there are many distinct regions involved in vision. And I think there are many interesting unanswered questions about how activity is coordinated across all of those regions. Historically, most recordings of the visual system happened one region at a time with only a small handful of neurons sampled in each study. And so what we set out to do was to capture activity of many of these regions simultaneously, both so we can survey their activity more efficiently and also so we can study how they interact with each other. So the Allen Brain Observatory wasn't the Institute's first foray into large-scale surveys. For about a decade before the Allen Brain Observatory launched, Allen Institute has been producing atlases of all kinds. And so, yeah, these would be systematic, standardized surveys of the mouse brain along different axes. So this was the survey that started it all, the Allen Brain Atlas. So this is a map of where genes are expressed across the whole mouse brain, which was really a transformative resource for the field. And that was followed by a map of connectivity between brain regions. So tracing where neurons, axons in a given region project to throughout the brain. And then more recently, Allen Institute has been doing surveys of cell types across the mouse brain and building up taxonomies of all these different cell types. Here shown cell types in cortex, but now this has been extended across the whole brain and also extended to human brain. So there's also a gene expression atlas of the human brain, and much more work now being done to kind of do comparisons across species. And this survey model has really become a cultural pattern at the Institute. And kind of Allen Institute is really dedicated to producing these big standardized, openly released reference data sets that then other groups outside the Institute can mine for their own needs. And so the Allen Brain Observatory was taking this idea and applying it to neurophysiology, but rather than mapping where genes are expressed or which areas are connected to each other, we wanted to map what neurons are actually doing in living brains. So here's what the platform looked like in practice. This shows 4 different facilities at the Institute, each optimized for a particular step in the data generation pipeline. So start with surgeries to prepare the animals. We use intrinsic imaging to locate visual areas, in vivo 2-photon imaging for measuring brain activity, and then last step is serial 2-photon tomography for slicing the brain after data collection is finished. And this level of standardization and specialization had not previously been attempted for in vivo physiology. We had dedicated operators for each step, surgeons who only do surgeries, experimenters who only run the rigs, because this specialization is critical for reducing variance. So for a statistics audience, I think this is a really important point. In a typical academic neuroscience lab, a single graduate student usually does everything from surgery to behavior training to recording to analysis. That's an enormous source of experimenter variance, and it makes combining data across labs or even across experimenters within a lab very difficult. So by separating out all these different roles and writing everything down as standard operating procedures, we were trying to turn neurophysiology into something closer to a clinical trial workflow where protocol is followed to the letter and the person executing it is ideally interchangeable with any other. So the first recording modality or recording technique that we used on this platform was 2-photon imaging. So in this case, we have genetically engineered mice whose neurons produce a fluorescent protein that gets brighter when a neuron is active. And so then you can measure activity across different neurons by measuring how bright they are at different times. So we use a specialized microscope that can look at the brain through a glass window, and then we record these movies of flashing neurons while the mouse is awake and viewing visual stimuli. And with 2-photon imaging, you can see a few hundred or a few thousand neurons per session with known spatial locations across multiple brain areas over multiple days. The trade-off is that the temporal resolution is limited, so what we see is a low-pass filtered version of the underlying electrical activity. And so this screenshot shows the results of this 2-photon imaging experiments, where this was the original web-based browser for this data set. So each row represents a neuron. Each column is a summary of how each of those neurons responded to a different class of visual stimulus. And in this first data release, this was actually kind of a revelation for us because we could put tens of thousands of neurons into this searchable, filterable interface, which is much different way of learning about the data than the kind of example neuron that you would typically see as a figure in most papers. So I wasn't super involved in the collection of the 2-photon imaging data set. My main focus was on another recording modality, which is electrophysiology. And for these electrophysiology experiments, we used what was at the time a brand new device called neuropixels probes. And development of these probes was funded by Allen Institute, along with Wellcome Trust, Gatsby Foundation, and Howard Hughes Medical Institute, and that the actual devices, the engineering was done by IMEC, which is a Nanoelectronics Research Institute in Belgium. And so NeuroPixels dramatically increase the number of neurons we can monitor simultaneously with electrophysiology. So this is the technique where you actually put an electrode right up next to a neuron and you measure its spiking activity extracellularly. So instead of looking at the brain through a window, you actually have to stick a device into the tissue, but the advantage is that you can monitor spikes directly rather than indirectly through their impact on these fluorescent indicators that you can see with 2-photon imaging. So on the left, you can see like the overall NeuroPixels probe package. The important part is, the the part that actually records from the brain is this narrow, thin shank, which is smaller than the width of average human hair, which you can stick into the brain, and there are more than 900 electrodes along the span of this shank, and you can read out 384 at a given time. And so this was many more than was possible previously with similar silicon probe recording devices. And so we could greatly increase the number of neurons we could record at once. So on the right, you get kind of a sense of the scale of this device relative to actual, well, not simulated neurons. So this is a rendering of a NeuroPixels probe penetrating through a simulation of mouse visual cortex. And you can see the size of the probe relative to the neurons that it's recording from. This particular slice of cortex is about a millimeter thick, and the whole mouse brain is maybe like 5 millimeters from top to bottom. And so with this 1 centimeter shank, you can record basically across the entire mouse brain at once. And because the overall package is so narrow, we can insert multiple NeuroPixels probes in different regions, allowing us to observe interactions between many different parts of the visual system. So this is a rendering of the rig that we built specifically for this electrophysiology survey. Put this up mostly to give you a sense of how much engineering goes into one of the experiments. Basically, all of this is custom designed for these experiments. The mouse sits, you can see like the little gray mouse in the middle of this image sitting on this wheel. Visual display is on the right. And around it, around the mouse's head are 6 micromanipulators. Each one holds an independently movable NeuroPixels probe, and they're kind of optimized so they can all descend into the mouse visual cortex. We also have cameras for tracking the mouse's eyes, body, and face, and hardware for synchronizing everything down to the millisecond. And we eventually built 5 identical copies of this rig, so anyone can, in principle, be swapped for another if needed. And yeah, this kind of standardization is really critical for generating reproducible data. And it's what allows us to maintain a steady pace of data collection if one of the rigs has to be taken down for repairs. So here's what the data actually looks like. On the left, you can see a cartoon of like where we're inserting our 6 NeuroPixels probes. So we have one targeting V1 primary visual area, and then the areas surrounding that are these higher visual areas which receive input from V1, and then further down, deeper inside the brain are these 2 thalamic regions, LGN and LP. As I mentioned before, LGN is what sends most of the visual input into V1, and LP would be considered a higher order thalamic nucleus because it receives input from V1 and sends it to these higher visual areas. And so, yeah, that's what the recording configuration is like. And then the actual data that we generate is, you can see on the right. So this is what's called a spike raster for about 10 seconds of data. The colors in the background indicate which area the spikes come from. And so each dot represents a spike. Each row represents a neuron. And so you can get a sense of kind of the high level patterns that you see in this data when you show different visual stimuli indicated by the gratings along the top. And so, yeah, the spikes that we're measuring are really the fundamental unit of neural communication. It's how information is transmitted across brain regions. And so this data at its core can be thought of as a high dimensional point process. And in this 10-second snippet, you can see very rich and different statistical signatures of these different areas. And so a typical session gives us spikes from a few thousand high quality neurons, and they're all sampled at sub-millisecond temporal resolution over more than 2 hours. So a lot of rich information in every data set. And because we're mainly recording from neurons in visual areas, they respond to what the mouse sees. So each session includes a diverse set of visual stimuli. These include artificial stimuli like gratings and also natural stimuli like natural scenes and natural movies. Um, so yeah, the artificial stimuli are useful because they're easy to analyze. We know exactly what the parameters are for each stimulus. But natural stimuli are more ecologically relevant and engage more engage the visual system in a more natural way. But maybe at the cost of being a little bit harder to characterize quantitatively. Um, so yeah, to keep things standardized, we show every animal the exact same stimuli in the same order. And these horizontal colored bars give you a sense of the timeline of one experiment. So the different types of stimuli are interleaved with each other. And actually the stimulus set that we showed for our NeuroPixels experiments, shown on the top with the gray icon, is basically the concatenated version of stimuli that were shown across multiple sessions for the two photon imaging data set shown in green on the bottom. So because with electrophysiology, we only get one shot to record from each neuron once we insert the probes during the experiment and then remove them once the experiment is finished. And so everything we're going to learn about those neurons, we have to learn within one session. But the advantage of electrophysiology is you can record from more regions simultaneously, and you can see their activity with very precise temporal resolution and read out the spikes directly. So I'll show some examples of how neurons respond to these visual stimuli. So drifting grading is one of the oldest stimuli in visual neuroscience. It's basically a set of black and white stripes that move across the screen in a certain direction. And by varying the direction and the speed of the grading, you can characterize what's called the tuning curve of the neuron. So in this middle plot here, you can see individual spike rasters for different directions and speeds. Speed is on the y-axis and direction is on the x-axis. And so within one of these boxes, instead of each row representing one neuron, each row represents one trial for the same neuron. So we're just looking at this neuron's response to a bunch of different stimulus, a bunch of different drifting gratings with different parameters, and looking to see how it spikes in response to these stimuli. And so I think it should be pretty clear from this that you get the densest spiking activity when the grading is moving to the lower left. So this kind of middle row, and you get a reasonable number of spikes regardless of what the temporal frequency or speed of the stimulus is. But you also see a fair number of spikes when the grading is moving to the upper right or 180 degrees off from that preferred direction. So that's a pretty typical response profile for a neuron to have one preferred direction, but also to respond pretty strongly at when the grading is moving in the opposite direction from that. And on the right, you can see this same data kind of summarized in this polar plot, which makes it really obvious which direction the neuron prefers. So this is the kind of measurement that visual neuroscientists have been making since the 1950s. But the difference here is that now, instead of recording from one neuron at a time, we're making the measurement across thousands of neurons simultaneously across multiple different regions in parallel. So a different and even simpler stimulus is these full field flashes. So these are cases where the screen is normally gray, but then it either turns white or turns black at random times with some interval between these flashes. And so on the right, you can see a spike raster for a single neuron to multiple presentations of both the light flash and the dark flash. You can see it responds much faster, much closer to the flash onset for the dark flash than the light flash. And this, even though this is a very simple stimulus, we can use it to probe activity in the visual system in a very standardized way. And kind of the latency and magnitude of responses to these different flashes tells you quite a bit about how that neuron responds to visual stimuli. And the last example I'll give is what's known as Gabor patches. This is basically just a drifting gradient with a circular mask, so it only shows up in one part of the screen. And then we present these circles at a grid of different locations all around the screen, just flash it for a brief period of time. And this allows us to map out the neuron's spatial receptive field. So this is the region of visual space where a stimulus actually drives the neuron's activity. So one important feature of visual processing is that each neuron's response is localized to a particular location in visual space. So for this neuron, it should be pretty obvious that the receptive field is basically in the middle of the screen, maybe a little bit slightly below the middle. But other neurons will respond to stimuli in different parts of the screen, maybe the corner or the edge. And some will have much larger receptive fields. So knowing a neuron's receptive field is kind of an important first step to knowing where in the visual scene it's actually processing information. So that's kind of the background for kind of understanding the content of the data set. What we actually released in October 2019, we called the visual coding data set because all mice were passively exposed to the same visual stimuli. So we're looking to see kind of what are the coding properties of neurons across the visual system without any regard to kind of what task the mouse is engaged in or what it's actually doing at the time. So the numbers here on the left give you a sense of scale of this data set. In visual cortex, we had more than 10,000 well-isolated units across these 6 different areas. The hippocampal formation, which happens to sit right between the visual cortex and the visual thalamus that we were targeting, that adds several thousand more. In total, we had data for around 40,000 high-quality units, each with spike times precisely aligned to all the same types of stimuli across 58 mice, which is an order of magnitude more data than what was collected in a single lab study. Now that neuropixels have proliferated, these numbers are maybe a bit less impressive, but at the time it was just the largest study of its kind. And the render of the right shows a dot indicating the spatial location of every neuron that we recorded. So you can see, the visual areas in the mouse brain are kind of toward the back of the brain, and we're only sampling from one hemisphere, but we're sampling very densely from these regions. And so we get a lot of data about the areas shown on the left. A few years later, in 2022, we released a second large data set, which we called visual behavior. So here, rather than just sitting and passively viewing all the different stimuli, the mice are trained to perform a task based on what they're shown. So this task involves viewing flashing natural images, and every time the identity of the image changes, the mouse has to lick. And if the mouse licks at the right time, when the image actually changes, then it gets a drop of water as reward. And so on the right, you can see here some example data across all these different areas. The spike raster, you can see that every time an image flashes, you get an increase in activity across many different areas. But especially when the image changes, you get this stronger increase in activity. And so there's a lot of neurons in the brain that are really tuned to this image change, because this is something that is very important for the animal in order to perform the task correctly. So this data set is super interesting if you want to study how neural activity predicts the animal's behavior on a trial by trial basis. So maybe some of you are interested in exploring this data yourself. So there are 3 different ways you can access it. First, we have a Python package called the Allen SDK. And if you are familiar with Python, this, I would say, is the easiest way to get up and running. So Allen SDK handles the download, caching, and local access of all the necessary files. So you can just focus on the analysis that you want to do. I would say this is what most users interact with, and it abstracts away a lot of the messy details of underlying file formats. But we also make the original data files available on the Dandy archive. So this is an NIH-supported archive for neurophysiology data. All of the data on Dandy must be stored in a Neurodata Without Borders format, also called NWB. So this NWB specification is designed specifically for storing neural time series along with other information you need for analysis, like the locations of the recording sites, the stimulus timing, etc. All of this is in a self-contained file. So if you're interested in doing analysis of spiking activity or two-photon imaging these days, you're probably going to have to get familiar with NWB, and Dandy is the main place where all these NWB files were stored. So you can also access the data as in the cloud. This is in a public S3 bucket on Amazon Web Services, which means that you don't have to actually download the data to use it. You can stream it directly from the cloud. And if in terms of the raw data that we collected, the entire data set is more than 100 terabytes. So you probably don't want to copy all that to your laptop. But having all three of these access methods is really essential for facilitating diverse types of data reuse, because some neuroscientists do still prefer to do their analysis locally, and we support that. But for kind of more high throughput machine learning workflows, you may already be running your analysis in the cloud. And so just attaching the data from a public S3 bucket is going to be a lot easier. So we had two publications describing these data sets. On the left is the publication from 2021, which described the visual coding data set. So if you're interested in this actual scientific discoveries that we made, I encourage you to check this out. And on the right is a publication for visual behavior data set, which is currently under review, but also available as a preprint on bioRxiv. That's definitely also worth reading, especially if you want to learn more about the data set in order to do your own analysis. But I think the more exciting story is that there are now well over 100 publications and preprints that have reused these data sets. So they come from labs all over the world, not just the Allen Institute, and they span a remarkable range of topics. One thing I found is it's impossible to predict in advance what the community is going to do with your data. A space of possible reanalyses for a data set this rich and this large is just much broader than we could have imagined at the time we were collecting it. So I think the best thing we can do is just make the data as accessible as possible and make sure everything is well documented and just get out of the way. So for most of these data sets, for most of these publications, the people writing the publication never needed to contact us. They just kind of use the publicly available resources to figure out how to use the data, and then went on to make discoveries with it. So the three main types of reuse that we see are one in the lower right cartoon is papers that directly compare our data with data from experiments carried out in their own labs. So this lets researchers perform an additional control using a completely independent data set, or test whether their findings generalize to a new context or a new brain region, or to a new recording modality. The second use case shown in the upper right is to provide the data needed to refine computational models, test new statistical methods, and refine biophysical models of the brain. Here, it's really important to have data on a large scale that's very standardized, so you can make sure that your results are replicated across many different subjects. And the third use case, shown on the upper left, is mining our data set for novel scientific insights. So these are papers that rely on our data to reveal new principles of brain function, and most of them focus on the visual system, but a handful look at other regions, such as the paper on the hippocampus I'll mention in a moment. So here's a nice example of comparing data with findings from another lab. This is a paper by Carson Stringer and colleagues in 2021. They wanted to understand how reliably a single neuron responds to a repeated static grating stimulus. So the original data set was based on two-photon imaging, but they wanted to make sure the results weren't just limited to that recording technique. So they decided to dig into our data, which included a very similar grating stimulus to the one they use, but in our case, all of the neurons were recorded with electrophysiology instead of two-photon imaging. And so these histograms show the distribution of trial-to-trial correlations across neurons, which look remarkably similar for the two techniques. So they were able to replicate their original result with data collected by a completely different lab with completely different recording modality. They didn't need to rerun experiments or they didn't even need to contact us. They just downloaded the data and compared it to theirs, and it really strengthened their findings. Here's a somewhat amusing example of data reuse for benchmarking methods. So in this paper, the author looks at correlations between firing rates of neurons in our data set and prices of Bitcoin and Ethereum over time. And obviously, there shouldn't be any neurons in the mouse brain tracking cryptocurrency prices, but when you run a naive correlation analysis, about 70% of neurons come up as significantly correlated. So the reason is that both neural firing rates and cryptocurrency prices are these slow-moving time series with strong temporal autocorrelation. And if you don't correct for that, you see these spurious correlations everywhere. So this is a really nice demonstration of a statistical failure mode that comes up all the time in neural data analysis. And when you have a data set of this scale, you can just apply the same analysis across tens of thousands of neurons and many sessions and show that this false positive rate is catastrophic. Third category of reuse is genuinely novel findings that we didn't anticipate. So these figures are from a study from Purandare and Mehta, published in eLife in 2023. So they're now looking at activity of neurons in the hippocampus while we're showing these natural movies. So hippocampus is a structure that's not traditionally considered to be a visual brain region. It's much better known for its role in episodic memory and its ability to precisely represent spatial locations. But surprisingly, they found that a substantial number of cells in hippocampus were spiking reliably at specific time points in this 30-second long movie. So here you can see raster plots for 4 example cells from the hippocampus, where the x-axis represents time within the movie or frames, 900 frames within the 30-second movie. And the y-axis are the 60 individual presentations of that movie. And again, each spike is represented by a dot. And so just naively, the responses of these neurons look very similar to what you'd expect to see if the animal was moving around its environment and you were measuring their activity at different spatial locations. So these look like what are known as spatial place fields of neurons, but they're actually the neurons responding to specific time points in this repeated movie. But the surprising thing is that these mice are head fixed. They're not moving at all. And they're not even trained to actively pay attention to the movie. So at least there's no reward for watching the movie. They're just experiencing passively. But regardless of that, the hippocampus clearly treats the movie as something important and learns to recognize specific moments in the movie in a highly reproducible way. So this definitely wasn't something we were looking for when we designed our experiments. And the fact that we recorded from the hippocampus at all was more an accident of anatomy than something intentional. And I think this is the category of reuse that's hardest to plan for, but most rewarding when it happens. And I think it's the strongest argument that we can make for open data in general. So in light of these data sharing efforts, there were some important lessons that we learned. First of all, it's clear there's a strong demand for highly standardized physiology surveys. When we started down this path, there was a lot of skepticism about whether the community would even be interested in something like this. Neuroscientists are very used to running their own experiments and wouldn't necessarily want to invest time analyzing data collected by someone else. but we found that in many cases, the consistency of a large-scale survey lets them answer different types of questions than they could with their own smaller, more variable data sets. It also lowers the barrier to discovery for people without access to their own data collection infrastructure, like statisticians. And we also happened to release our data in 2019, right before the pandemic. And when many labs shut down, people had extra time on their hands to start mining our data, so they could continue to make discoveries even when they couldn't run their own experiments. So second, we learned that facilitating different types of reuse is essential. Some people want to analyze pre-computed metrics across the whole data set. Others want to dig deep into individual sessions. Others want access to the raw data. And we need to be able to support all these different access patterns. Third, it's clear that standardization of protocols, file formats, and metadata is a huge force multiplier for reuse. It takes a lot of effort to dive into a new data set. And the more people can rely on things that they already know, like APIs that they're already familiar with, the more they will be willing to commit to this analysis. And yeah, knowing that all the data from the Allen Brain Observatory follows the same conventions was a big plus for convincing people to reuse it. And then fourth, I think we're seeing that cloud-based analysis is becoming increasingly important as the scale of data grows. If you want to access the raw data, it makes way more sense to run your analysis where the data already lives rather than trying to copy it to your local infrastructure. So I think, yeah, cloud-based data sharing is really the future. So with those lessons in mind, I want to talk about some promising developments that have happened since our initial data release. So the first builds on the fact that people are eager to analyze highly standardized physiology data, but what if we can give them more control and let them propose the experiments to run on our rigs? So that's the idea behind OpenScope, which is organized like a telescope time allocation program. So you have outside researchers now submit proposals, and as long as those proposals fit within the capabilities of our rigs, like using visual stimulation and recording either with neuropixels or two-photon imaging, those proposals can then get run by the Allen Institute on our rigs with our operators using the standardized protocols that we've developed. And after that, the teams get a year to analyze the data on their own before it's made publicly available. And this has turned out to be enormously productive. It means that the platform can be used to ask questions that we wouldn't have thought of, and it means that people who don't have access to their own NeuroPixels rigs can still test their specific hypotheses with experiments that they design. So one example of an OpenScope project, which resulted in a recent paper by Shin and colleagues based at UC Berkeley, is focused on illusory contours. So you've probably seen something like the figure on the left, which has these three Pac-Man shapes arranged in a way that makes your visual system perceive a triangle in the middle. If you stare at it, it really looks like there are edges of the triangle, but of course they don't actually exist. They're completely constructed by your brain. And so to understand how the mouse brain processes illusory contours, the team developed a set of stimuli shown on the right, where in the top row, you have these actual illusory contours. So there's not actually a real bar going across, but in the bottom row, you have the same orientation of the bars, but in this case, they do have real edges. And so they first showed these stimuli to mice in their own lab using two-photon imaging, and they focused on neurons which were processing information exactly in the center of the screen, right where this bar passes through their receptive field. And they found that for the bars that had actual edges, the neurons had a preferred tuning direction, just like the preferred direction that I talked about with the drifting gratings. But remarkably, they found that those neurons also responded very robustly to the stimuli with the illusory contour, but only when that illusory contour was at that same preferred orientation. So these neurons are clearly responding to the presence of a contour, even when the information in their receptive field is just completely blank. And so then they turned to OpenScope and proposed running the same experiment, but with neuropixels. And using the NeuroPixels rig at the Allen Institute. They got basically the same result. So this is showed by the red along the diagonal line, showing that the response of the neurons to the stimuli with real edges is basically the same as the response of neurons to the stimuli with no edges. So clearly showing that these neurons are processing illusory contours, and then they go on to talk more about the mechanisms by which this might happen. And so beyond OpenScope, another exciting development is that sharing large-scale electrophysiology data is becoming much more common in general. So a number of large, high-quality, openly shared data sets have come online in recent years. This slide shows 6 of them. Here again, each dot represents the location of one neuron within the mouse brain. All of these data sets are available on the Dandy archive, which I mentioned earlier. And yeah, each data set represents kind of an overlapping but different, distinct set of brain regions. So yeah, a few years ago, if you wanted to develop a new method for neural data analysis, you could maybe benchmark it on one data set you could easily access, but now you can download all of these data sets, some of which include more than 100,000 neurons, all of which were collected under different conditions, and kind of see if your analysis method generalizes across all of them. In particular, I want to highlight the International Brain Laboratory dataset, which is the one in the bottom row in the middle. This is output of 10 different labs doing standardized recordings across the whole brain in mice performing the same task. And so it's very much in the spirit of Allen Brain Observatory, but it's being done across institutions rather than within one. So yeah, I want to finish by talking about some recent developments at the Allen Institute. In the years after our 2 big data releases, we launched a new scientific division called Neurodynamics. And this is where I lead the electrophysiology group. Our focus has shifted away from the visual system to covering pretty much the whole brain. And so we've developed a lot of new tools like this new rig shown on the left, which can insert more than a dozen NeuroPixels probes from almost any angle. And we're also using MRI to scan the brain of each individual mouse before we record from it. So that lets us target deep brain structures much more reliably than we could before. And of course, we're carrying forward our commitment to open data that define the Allen Brain Observatory, but we're doing things a little bit differently. So first of all, we want to make the data public as soon as it's collected. So instead of storing the data on our local servers, the data is immediately uploaded to a public AWS bucket the same day that the experiment runs. So that means that our internal data analysts are working on the same public resource that outside scientists would be. And so this also means that once the data leaves the rig and goes to the cloud, all analysis happens in the cloud. So we want to be bringing people to the data, not the other way around. If you want to work with our data sets, you can spin up a compute environment that's already connected to the data without downloading anything and get up and running much quicker. And we recommend platforms like CodeOcean that make this cloud-based analysis much more accessible for scientists. And third, I think this is the most important conceptual shift, is that we still want to put a lot of effort into data sharing, but here data sharing is primarily about documentation and curation rather than uploading. So the work of data sharing is making sure everything has rich metadata so people can interpret it, the right indexing so people can discover it, and good documentation and tutorials so people can actually use it. This whole framework is a big credit to David Feng, Saskia DeVries, and our scientific computing team, who've really honed this philosophy and built out our data sharing infrastructure. So this slide shows our 4-pillar data sharing framework. Accessible means that data lives from the start on AWS Open Data Bucket. All of our code is in public GitHub repositories, and all of our protocols are published on protocols.io. And so, yeah, standardized means that we've adopted community file formats like Neurodata Without Borders for physiology, OMEs for imaging, and our own open source schema called AND Data Schema for metadata. Transparent means there's a searchable metadata catalog, essentially a big JSON document database, which you can query to learn about what's in our experiments. And this will also be complemented by data portals where you can see kind of quality control visualizations for every experiment. So you can know exactly what a session looks like before you invest time in analyzing it. And reproducible means that we use CodeOcean to run our analyses in a way so that all of the inputs and outputs are tracked. And yeah, that makes it much easier to replicate our results because you're not, you can just share a link that includes the code, the computing environment, and the data rather than having to track all these things separately. So overall, this is a big commitment of engineering effort, but I think it pays off every time someone wants to reuse our data or extend our analyses, which they can do much more efficiently now. And so last thing I want to talk about is how we have to adapt our approach given the rise of AI. So all of the infrastructure I discussed was developed before we knew how much of an impact AI was going to have on science. And I think if there's ever a time to rethink how and why we share our data, that time is now. So first of all, I think standardization we knew was important, but now it is more important than ever because large language models are extremely powerful at navigating well-structured data, but they stumble or could even hallucinate plausibly when the structure is inconsistent. So every idiosyncratic file format, every undocumented convention is going to be friction that the next generation of data analysts are really going to have to pay for. Second, it's clear that writing analysis code is no longer the bottleneck that it used to be. If you have a well-documented data set and a clear question, you can often get to the first analysis within minutes using AI assistance. But now this bottleneck shifts to setting up the right context. You really need well-curated metadata, good documentation, clear schemas to let an AI assistant or any analyst, human or otherwise, actually do useful work with the data. So we need to invest much more time here, building learning resources that both humans and large language models can easily understand. And third, I think code reuse and provenance tracking have always been a problem, but I think they're a much bigger challenge now than they were before. When a human researcher is writing analysis code, there's a strong incentive not to reinvent the wheel, if only to save time. Although, of course, lack of discoverability of code still leads to a lot of redundant development. But when you tell an AI assistant to write your analysis code without specifying any details of the implementation, it can very quickly generate a lot of code from scratch that doesn't rely on any existing libraries, and this code will be much harder to validate. You can ask the LLM to do it for you, but that's often not a priority if the answers it's spitting out seem to make sense. So overall, the barrier to writing code has collapsed, which means the barrier to writing throwaway code has collapsed too. So for a field that cares about reproducibility, I think that's a huge problem. If everyone is now generating thousands of lines of new code for every analysis, the validation becomes extremely difficult, even if the underlying data is the same. So I think overall, AI creates a huge opportunity for increasing the pace of a discovery by allowing scientists to do more complex analyses faster. But we need to be very careful about how we use it, or else we will exacerbate many of the problems that already exist in the space. I think the collaborative data science infrastructure we're setting up at the Allen Institute is designed to address exactly these problems. And I'm optimistic that we can update our strategy to respond to the latest developments in AI. But yeah, ultimately, data sharing is necessary for taking advantage of the rapid pace of progress, but it's definitely not sufficient. So I hope people will pay attention to some of the lessons that we've learned in the course of collecting and sharing our large-scale physiology data, which are even more relevant in the age of AI. So I want to end by thanking all the people who contributed to the work that I discussed today across the Allen Brain Observatory, OpenScope, and Neural Dynamics teams. There are many more contributors than I'm able to list here. But yeah, my favorite thing about working at the Allen Institute is being able to collaborate with experts across many different domains. And that makes these ambitious data collection efforts possible in the first place. And I also want to thank everyone who's reusing our data. I'm consistently surprised by the discoveries that continue to emerge from the Allen Brain Observatory data sets, which go far beyond anything we initially expected when we started this work. So thanks to everyone for watching. I think we have time for a few questions.